Hi, I am Gianna Ship, and today my group and I we are presenting on the cell cycle and cancer. I'm going to let my fellow group members introduce themselves.
Hi, I'm Catrice Cook.
Hello, I'm Ashley Gill.
Hi, I'm Chanel Carrington.
Hi, I'm Hillary Mosley.
Okay, so today, as has been mentioned already, we will be discussing the interdependency of cell cycle, cancer, and your genetic makeup. It's important for us to look at cancer because it affects people each and every day. We will begin by discussing the cell cycle. And before we begin, it's just important to say that the MAP kinase pathway is actually the signaling pathway that initiates cell cycle. And Ashley will discuss that later on in our presentation. But first, Chanel's going to give us a discussion of the phases of the cell cycle.
The cell cycle is a series of where the cell goes through division and replication. The cell cycle begins where two new cells are produced by division of a single parent cell and ends with one of the cells divided again into separate into two cells. The four phases. The first phase is G1 phase, which is the longest phase into interphase and occurs before DNA synthesis. G1 phase is in charge of making a very important decision in the cell cycle. It is charged of determining whether the cell cycle will be able to divide again. S phase is the second phase where the DNA replication begins. Throughout the cell, the growth continues through S phase as well as the rate of synthesis of a number of proteins and enzymes that are involved in DNA synthesis. G2 phase, which occurs right after DNA synthesis. In G2 phase, all of the organelles inside the cell are replicated in order to prepare of mitosis. Cells have one last chance to produce more proteins in the G2 phase before division. And M phase is called mitotic phase, which consists of two main parts, mitosis and cytokinesis. The results of M phase is two daughter cells. The first part is mitosis, which includes 5 phases, prophase, prometaphase, metaphase, anaphase, and telophase. Telophase is immediately followed by cytokinesis. In prophase, chromosome material is condensed to form mitotic chromosomes. Disassemble the cytoskeleton. Mitotic spindles are assembled and the nuclear envelope dispersed. In prophase, there are 92 chromatids and 46 chromosomes. Next is prometaphase, where the chromosomes are being attached. When the cell enters prometaphase, it is the nuclear envelope that breaks down and the chromosomes are attached to spindles, microtubules, and move to the spindle equator. Once the chromosomes are spindles, microtubules reach the equator, the cell enters metaphase. After that, there is metaphase, where there are 46 chromosomes and 92 chromatids. In metaphase, there are fully condensed chromosomes that are aligned at the metaphase plate attached by chromosomal microtubules to both poles. Besides that is anaphase that are characterized into two kinds of movement, anaphase A and anaphase B. During anaphase A, chromosomes and centromeres split towards the spindle poles and connect cores. Microtubules get shorter. In anaphase B, the poles move away from each other and microtubule lengthen. Anaphase A and B occur at the same time. By ending the anaphase, there are 92 chromatids and 92 chromosomes. Finally, there is telophase, where there are 92 chromatids and 92 chromosomes in telophase. Once telophase Once in telophase, chromosome clusters at opposite spindle poles. The chromosomes begin to disperse, and the nuclear envelope assembles at the organelles form. Telophase is completed. The cell leaves M phase and enters cytokinesis.
Now that we have an overview of the cell cycle,
it's important to know the checkpoints and where they are found in the cell cycle. So we need to know where they are, what do they do, where are they, and essential, what happens if they don't pass the checkpoint, and what happens if they do pass the checkpoint. All right, so we'll start off with what is a checkpoint. A checkpoint in the cell cycle is basically a control system that monitors the cell cycle. Leland Hartwell and Ted Wiener proposed the theory of checkpoints because they knew that something was responsible for regulating the cell cycle. There are 3 functions that checkpoints do depending on their location in the cell cycle. All right, which brings us to the checkpoints and their locations. The cell cycle is split into 2 major components, interphase and mitotic phase, also called the M phase. During interphase, there are 3 phases, G1 phase, S phase, and G2 phase. And then during M phase, it consists of 4 phases, prophase, metaphase, anaphase, telophase, and then it follows up to cytokinesis. So where are these checkpoints? The picture on the left shows where each checkpoint is located. The first checkpoint is at G1, the second one, G2 phase, and then the third checkpoint is at the mitotic phase. So the first checkpoint is in the G1 phase. It is found at the ending of the G1 phase. What occurs during this checkpoint is they check for nutrients, cell size, growth factors, and DNA damage. However, if there's something goes wrong, such as the nutrients wasn't available or the cells aren't ready to proceed on, they go into the G0 phase. This is called the resting phase in the cell cycle. Cells enter the G0 phase because they are not ready to divide. Cells can stay into the G0 phase for hours, days, months, or even years until they are ready. And if they never are never ready to divide, they will die. Which then brings us to the G2 checkpoint, well, at the S phase. You move to S phase, and then once it moves from S phase, it goes to the G2 checkpoint. So the G2 checkpoint checks for the cell size and DNA replication. Once that passes the checkpoint, it moves into the M phase. So, as Chanel stated previously, everything that happens during the M phase, it goes to the checkpoint. The M checkpoint is responsible for making sure sister chromatids attach to the spindle. The picture on the bottom right shows the spindle as well as the sister chromatids, which the sister chromatids are found as marked as the red X's. Which brings us to CDKs and cyclins.
Okay, so I will be discussing the CDKs and cyclins. To begin, the reason why we're talking about this next is because CDKs and cyclins are the mechanisms by which these checkpoints are able to work. And now I'm going to go back to the presentation. Okay, CDKs and cyclins. So cyclin-dependent kinases. These are protein kinases that help with the progression through the cell cycle. CDKs are actually produced by the CDC2 gene when they're encoded. And they are given this title because CDKs only become activated when they're bound to an activated protein called a cyclin. There are multiple types of CDK cyclin complexes, and they vary based on the part of the cell cycle they control. And that also is how they earn their name. There's 2 examples, the S-CDK cyclin complex and the M-CDK cyclin complex. The S-CD cyclin complex is responsible for regulating the licensing of DNA, which is basically the process that ensures DNA molecules are not replicated more than once. This CDK is produced at the start of the S phase and activates DNA replication at the locus where the licensing is occurring, and then also prevents relicensing. It's important to prevent it so that, as said before, the DNA is not replicated more than once. But what makes the CDK cycling complex really unique is the fact that they are able to function at the proper moment that they're needed. This image shows us different methods by which the CDKs are regulated. The first one is the association with the cyclins, which I mentioned just before, is that CDKs do not work until they bind to cyclins. CDKs and cyclins are also regulated by phosphorylation and dephosphorylation. In order to activate a CDK cyclin complex, a CDK threonine residue must be phosphorylated, and this is catalyzed by the CAK, the CDK activating kinase. And this is also, there's also the CDK inhibitory phosphorylation of the threonine 14 and tyrosine 15. There's also association with CDK inhibitors, which are also called CKIs, and they monitor the activity of CDK cyclin complexes by binding directly to them. Another way that we regulate how CDKs and cyclin works is through the cyclin regulation by concentration. So the concentrations of CDKs remain constant through the cycle, but cyclin concentrations fluctuate. So the example that I mentioned earlier is a great way to describe this, which is the mitotic cyclin CDK complex. The cyclin concentrations gradually increase as they reach a specific phase. So for mitotic cyclin, the threshold is reached at the G2 phase, so it allows it to activate the mitotic CDK when it binds. But after it's used, the cyclin is degraded, and this is done by ubiquitination. So ubiquitination binds the cyclin to tag it for degradation, and the cyclin is degraded by proteolysis, and that is ubiquitin dependent. So basically, when the ubiquitin is identified as being attached to the cyclin, proteolysis, which is the breakdown of proteins into polypeptides and smaller amino acids, occurs. And next, I have an image that basically sums up the checkpoints that Catrice discussed and the CDKs and cyclins that I went over. So basically, as you see, each cyclin and CDK complex correlates to a different checkpoint throughout the cell cycle. And they are, as I discussed before, done by the phosphorylation, which involves ATP and ADP. And then you also can see the degradation of the cyclins throughout the whole entire cycle. It's just a good photo that explains everything. Next, we have the discussion of the deregulation of the cell cycle.
Okay. All right. I'll begin discussing the deregulation of the cell cycle. One of the pathways is MAPK pathway. This pathway regulates the cell cycle and activates genes found in the cell cycle. Now I have a picture that will show you a good description of the MAPK pathway. This pathway is activated when a specific ligand known as the epidermal growth factor, or EGF, binds to its receptor. Once the receptor is phosphorylated, signaling molecules such as GRB2 and SOS are recruited to the internal site, resulting in RAS activation in the membrane. The scaffolding proteins regulate the effectiveness of signal transduction, known as kinase suppressor of RAS, also known as KSR. RAS triggers the phosphorylation cascade involving RAF, MEK, and ERK, leading to ERK captivation of the nucleus. Once ERK is activated, there are several transcription factors, such as JUN, which intervene gene expression. These target genes that are activated by MAPK signaling contribute to high proliferation, resulting in exponential growth potential causing cancer.
A proto-oncogene is a normal cellular gene that is involved in cell growth and proliferation. They can be mutated to a cancer-promoting oncogene. They are mutated by changing their protein coding section and by altering their expression. There are 7 different proteins that can control cell growth. They are growth factors, growth factor receptors, signal transduction proteins, transcription factors, pro or anti-apoptotic proteins, cell cycle control proteins, and DNA repair proteins.
The mutations changing these structures or proteins expression of these proteins generally gives rise to dominantly active oncogenes. An oncogene is a gene that in certain circumstances can transform a cell into a tumor cell. One example of an oncogene is ERBB2, which encodes for breast cancer. Another example of a proto-oncogene is Ras, which normally encodes for intracellular signal transduction proteins, but the mutant form Ras-D becomes responsible for the uncontrolled growth promoting signal. And also the proto-oncogene MYC encodes for transcription factors. When it becomes mutant, it is responsible for increased production rates and gene expression is altered, giving rise to tumor formation. And there are quite a few ways that proto-oncogenes can be converted into oncogenes. One of them is point mutation, which results in constitutively acting protein products. Localized reduplication, which leads to the overexpression of the encoded protein. And chromosomal translocation, which causes the growth regulatory gene to be under a different promoter, and this causes inappropriate expression of oncogenes. And now Octavia will give you a more in-depth discussion of the different types of conversions from proto-oncogenes to oncogenes.
Okay. This type of is application and the first type is application. Okay. When the number of copies is all the protein, the total is the absence of protein. The next type is type mutation. It's a first way for protein pathway to be leading to excessive cell chromosomal amplification, a portion of the chromosome is translation of oxygen might be moved from chromosome 8 to 14, where it's situated to the active protein clothing and molecules. Means the MYC goes to the active active. Involved in the space of protein is a new genetic material into a normal gene, particularly a into chromosome where total oxygen is located. Next, we'll be talking about tumor.
So it's significant to know what are tumor suppressors. You know what they do and where they are found. So let's talk about that. Tumor suppressor genes. The function of tumor suppressors is to restrain cell proliferation and are found in the cell cycle. There are 5 broad classes. These are 1, intracellular proteins, 2, checkpoint control proteins that arrest the cell cycle, 3, enzymes that help with DNA repair, 4, proteins that promote apoptosis, and 5, receptors for secreted hormones. These 5 classes are the reason for how and why the cell cycle operates. So let's look into some examples of tumor suppressor genes. There are many tumor suppressor genes. The major tumor suppressor is retinoblastoma, known as RB for short. RB is a tumor suppressor gene whose function is to block E2F, a transcription factor. Retinoblastoma is relevant to the cell cycle because it controls progression from G1 phase to the S phase. And then it's critical to know what happens if a tumor suppressor goes wrong. So let's talk about mutation of tumor suppressor genes.
When determining a cell if it has become inactive, the scientists perform a cell fusion experiment. Tumor suppressors are normal functioning is to restrain cell proliferation, which is basically a break in the process. If there is no outcome of an inactivation of the p53 pathway, this will lead to failure of apoptosis. This will then contribute to the development of cancer by allowing the survival and reproduction of the cell to contain damaged DNA and not allowing the genes to perform their functions properly. P53 genes code for p53 protein, which is their response to damaged DNA. The proteins can either have a repressive or inhibit effect on the regulation of the cell cycle, or can either promote apoptosis. Yeah. If there is a repression of the genes, the cell cycle will not continue to express the gene, and this will inhibit the cell division. Since these DNA cells are unprotected to different agents that causes DNA damage on a DNA cell, it causes the cell to stimulate the p53 pathway.
Now begin talking about the multi-step process. The first step is tumor initiation. This is based on DNA mutation. The second step is tumor promotion. The initiated cells are stimulated to proliferate. And then the last step, which is called tumor progression, this is where tumor cells' properties progressively change over time as the cells obtain more abnormal traits and become aggressive. Now I'm about to show you a picture. Okay. Next slide. Tumor promotion is the process where the initiated cells become expanded into papillomas or polyps, one of which can act as a promoter for the ultimate development of cancer. These essential proliferations are the immediate indicators for promotion. These compounds promote the proliferation of the cell, reproducing a large number of daughter cells containing the mutation created by the initiator. Promotion then stimulates the mutated cells to divide and form tumors. Promoters do not conveniently bind to DNA or macromolecules within the cell like the initiators do. Instead, several bind to receptors on the cell surface in order to influence intracellular pathways that lead to increased cell proliferation. The last step of the multi-step process is tumor progression. During this progression, tumor cell properties progressively change over time as the cells attain more abnormal traits and become gradually more aggressive and advanced. This step shows the continuation of additional mutations occurring within the cells of the tumor population, causing cancer.
So we've discussed several things about the cell cycle and cancer and genetics. And so now we want to discuss how these things are interrelated and how they kind of apply to life and what their implications are. We'll go back to the presentation. Okay, so implications and conclusions. Epigenetics. There is a thing such as cancer epigenetics, and that is the study of epigenetic modification of the genome. And this basically causes the development of cancer cells. And these mechanisms of cancer genetics are DNA methylation, which is an example, and it causes the silencing of tumor suppressor genes. And what's interesting, through research we found that the inactivation of tumor suppressor genes occurs just as often as inactivation by DNA mutation, which proves that epigenetics is a very significant method that alters gene expression and influences cancer. Individuals can have changes in their DNA from other things, such as changes in the regulatory protein, such as WEE1. And that has actually been researched as a possibility to treat cancer. Actually, in 2014, in March of 2014, researchers identified WEE1 as a target for treating medulloblastoma. And this was performed through an integrated genomic analysis, which involved the use of siRNAs and the profiling of patient samples through cell cycle related kinases. So that just shows why the interdependency of cell cycle regulation and its checkpoints are important. Also, we just want to add that, so we want to just go ahead and give a summary of everything we've discussed today. So today we discussed that cancer, that the regulation of the cell cycle and the checkpoints enable a cell to function and divide efficiently, but when it doesn't happen, the cell cycle can be deregulated. Cancer is also caused by mutations at the genetic level, such as proto-oncogenes and tumor suppressor genes. And when these mutations accumulate, cancer does occur. Studies are constantly being done on the genetic and cellular factors that influence cancer, and that helps us better understand the disease and improve treatments. Thank you. Have a nice day.
Bye.